reconstruct the patterns along which the viruses spread and transmit. So, and before I get there, because these tutorials and sort of practicals tend to sort of fizzle out in the end, I'd like to make sure that we acknowledge, everybody who has contributed to this effort over the years. So as I said, sort of Next Strain and the kind of ecosystem of things I work in has been a terrific collaboration with Trevor Bedford in his lab at the Fred Hutchinson Cancer Center in Seattle. Over the years, there have been a large number of people involved, and particularly in the context of this this pandemic, we have worked closely with James Hadfield, sort of Emma, who's also one of the instructors, Ivan Axamentov, who is a software developer in my lab, Maura Zuber, Tom Sibley, and others. Importantly, also the data that we are going to discuss and use here and analyze are not data that we ourselves sort of collected, but these are data that scientists, clinicians, healthcare professionals around the world have collected, sequenced, and made publicly available, often through the data sharing platform GISAID, for us to for everybody to analyze and use this to fight this pandemic as well as we can. On Anya's side, there's also a large number of people involved in the tools that she's going to present. There is a crew at the University of Edinburgh, sort of led by Andrew Rambo, Eddie Holmes from Sydney, the folks from Oxford, and so forth. Much of this is also is implemented by the Center for Genomic Pattern Surveillance in London. Okay, but now let's get going. So when you typically think about an outbreak of a pathogen of an infectious agent, you sort of start with some index case, and you get sort of transmissions, and you get this tree of transmissions, which is sort of expanding in time. And most of this is not actually observed, especially with SARS-CoV-2. By the time we observed these outbreaks for the first time, we typically already had a big outbreak ongoing. So there's only a small fraction of these cases that end up being sampled, right? Now, sort of as the virus is spreading and replicating its genome, it also sort of tends to mutate once in a while, sort of copying mistakes, sort of typos when replicating its genomes. And these mutations are inherited by all descendants of this particular event, of this particular transmission. Now, this is sort of where then the sequencing and the phylogenetic reconstruction comes in. You can clean this up a little bit, and you get a phylogenetic tree, which essentially is some sort of family tree of the virus. And this is what, just a big picture view of what we'll sort of try to get at over the next hour or so. So just to summarize this, slightly, so we are sort of dealing with this kind of transmission tree. We have transmissions, which are the red dots here. There's mutations that happen somewhere along this transmission process as the virus is replicating. A minority of the cases is sampled, and this sort of ends up being these sampled cases. They sometimes will not only be sort of PCR assessed, but also people will go through the extra effort and sequence the genome, meaning that 30,000 string of ACGT, or RNA in that case, letters, which then needs to be analyzed. So what kind of data are we dealing with when we look at sequence data? This typically, once you get it, will look something like this. So you have what's called sort of a FASTA file, where you have a header of a genome, of each of the sample has sort of a header, and then there's a sequence, which is just a string of ACGT, sometimes sort of other sort of codes that stand for ambiguous sites or missing data and things like that. But looking at this like this is sort of basically alphabet soup, right? So this doesn't really give us any insight in what's going on. And the first step that we need to do to meaningfully analyze these data is to align the sequences. Meaning we have to find an arrangement such that sort of corresponding letters are on top of each other, that they are in the same column. And this, for this particular case, would sort of look a little bit like this, where if we, for example, shift this sample B by 3 letters to the right, we'll see that we get sort of a correspondence here between bases. It just ends up being a little longer now with 3 insertions here. And similarly, these other 2, they need to be shifted by 2. And here we actually have a little piece missing where we need to, that could be a deletion, where we need to introduce 3 gaps here to make this match up both on this side of the gap and on that side of it. So this is a process sort of called sequence alignment, and what it's sort of fundamentally doing, it's sort of trying to arrange sequences such that homologous parts of the sequence, kind of corresponding parts of the sequence, end up being on top of each other, and that you can meaningfully sort of look at the changes in these data. Luckily, there's a large suite of third-party programs out there that do this for you. One of them is called MAF, there's Cluster W, there's Muscle. These programs are also, and alignment algorithms are integrated in sort of more, point and click editing, genomics applications like Genius and so forth. So this typically isn't a problem that is a big obstacle, but it's something that needs to be done before you actually kind of start using these data. Now, an alignment would then typically be viewed in an alignment viewer, which looks something like this, where just helpfully here, the different letters are colored, A green, blue C blue, black G, T red. And you see, for example, here in the middle, in this part, so each row is 1 genome. In this case, the flu genomes, but the principle is the same. Here we have a deletion that these genomes here, they are missing 3 amino acids or 9 nucleotides in this position. You also see the odd mutation here. So in this column, for example, pretty much everybody has a T, and then there's 1 C here, and then we start with T. And you see these mutations so sprinkled across this alignment, so tiny little sort of single nucleotide differences between genomes that we will use. These are precisely the ones that I showed you on the transmission tree before, and these are the ones that we will use to reconstruct phylogenies and sort of the relationships between different viruses. Now, this is precisely the process that I've sort of summarized here. So what I've showed you in the previous slide is an alignment, and this is kind of a stylized sort of miniature, comic version of this is shown here, where each genome is one of these black bars, and the diamonds here, the colored diamonds, would be mutations which are shared here by 5, by 2, by 4, and so forth. And we can now, kind of almost by hand, sort of reconstruct a phylogeny, right? So these samples A and B, they share 3 mutations where A has 1 extra one, so they're probably pretty closely related. So we'll sort of put them together first. Then they share the cyan one, which is not shared by C and D. So there might be sort of a link like this, where D has 1 extra mutation, the blue one, and so forth. So you essentially can, for at least this simple example, you can do this by hand to sort of get this idea. In practice, this is a more complicated problem for which, again, there is quite a bit of third-party software available that we and others use every day to do these kind of analysis. But hopefully you see that there is some sort of there is information in sequence data like this about the relationship between these samples, which then again gives you some information about the transmission process that is the thing that you're actually interested in. And the relationship between these two, the uncertainty, the kind of thing that you can conclude, is something that we'll cover over the next 45 minutes or so. The software that you typically use to do these kind of analysis are called IQ-Tree or RAXML is the popular one, FastTree. And these kind of software are the ones that underlie the dynamics that we'll show in a second. Okay, then sort of getting to the tree, to a real tree, not like a toy tree that I showed you before. They are typically displayed like so. This is a nice sort of colorful example of, in this case, a Zika tree from NextStrain. And I just want to use this to introduce a bit of the terminology. So in this case here, the tree goes from left to right, whereas the ancestral variant, so the ancient, the precursors, they are on the very on the very left. And in this case, this is a common ancestor. This happened about 2006, while the present day variants are on the right. And as you go from right to left, you sort of go back in time, or in kind of mutation and ancestry and mutation space, where we'll often refer to the actual sample. So these little circles at the tips of the tree, we'll refer to them as tips or as leaves. Then the parts that connect different bits and pieces of the tree, they're called branches. And the branch points inside the tree are often called null nodes. So this here would be sort of a rooted and timescaled version of a tree. Sometimes you also come across other representations of the tree where you don't have sort of a specified origin or root, but you'd rather sort of have a more agnostic approach, sort of just wanting to show differences between trees, and you'll find something like this. These are the same trees, just displayed in a different fashion. So this is just to avoid being, the fact that there's multiple different ways of looking at a tree should not confuse you too much. Good. So that was the first little bit on the very basics of phylogenetics and epidemiology, molecular epidemiology. What I want to do in the next few minutes is sort of recap the broad, in very, very broad strokes, the evolution and spread of SARS-CoV-2 over the past few months, and then hand over to Emma. So yeah, so that is the next presentation that we prepared. So as you of course all know, sort of SARS-CoV-2 was first described in late December last year, and then the genome was published and shared just a few days later in early January. And we've come a long way since. So what this early genome told us, that we are dealing with a beta coronavirus, which is a reasonably close relative of SARS-CoV-1, so the 2003-2004 SARS outbreak, but that is part of the family of beta coronaviruses, where we also have two seasonal cold viruses, the human coronavirus H343 and HKU1, as well as MERS. So now zooming into SARS-CoV-1 and SARS-CoV-2 a little bit, we see that it's sort of embedded in quite a diverse sample of genomes that are mostly sampled from bats or pangolins. But there's no, and Anya will talk a little bit more about this later, there's no sort of immediately close ancestor that we know in the animal reservoir. Now, we've been tracking this diversity of SARS-CoV-2 since the very beginning, and at the very beginning, things looked more or less like this. So this is a phylogenetic tree. It doesn't look like much like a tree, but this is a state of affairs on January 26, when we had on the order of 20 genomes, and quite a big chunk of them actually sort of ended up being at the root of the tree, at the very left, as I just discussed, right? And these are genomes that are all identical. They are, on all the 30,000 bases of the genome, they are letter to letter identical to the reference sequence, to the sort of prototype sequence from Wuhan. And then there's a few sequences that are 1 mutation away, some that are 3 mutation away, and there was the odd one that was 5 mutation away. So that was in the very beginning. And since then, sort of in an unprecedented sequencing effort has been launched, and we've seen an enormous increase of genomes over the past 8 months or so. We are now at sort of more than 100,000 whole SARS-CoV-2 genomes, more than anybody can analyze and use, really. And that's why it's all the more important that we sort of develop tools to help people around the world to analyze, and zoom in, zero in on their specific questions that they have with genomes in their communities. So, the current overview of the SARS-CoV-2 tree looks roughly like this. So this is again sort of the root sequences, the sequences that circulated in Wuhan at the very beginning, and from there the virus has been mutating, as all the RNA viruses do. That's a very common feature. which is not terribly remarkable, even though it's discussed in the media often in quite sort of alarmist terms. So we've observed that sort of with every month, we find about, on average, 2 additional mutations in the genome of SARS-CoV-2 samples. And these are visible here as these sort of vertical lines, which correspond to the discrete number of mutations by which these genomes differ from the original Wuhan strains. This sort of gradual increase of mutations you see here, where on the y-axis we plot the number of mutations against time, which has sort of a fairly regular, if noisy, increase in the number of mutations. Good, and yeah, so this is just sort of a rough breakdown of the clades, about which we'll talk a little bit further, and maybe I'll just hand over to Emma now to show you how to explore these data in sort of real interactively by yourself in NextStrain.
Yes, happy to take over. Okay, so let me share my screen. So as Richard says, I'm going to talk through kind of using SARS-CoV-2 as a specific example and looking at what we can actually see and how we can manipulate NextStrain, because it's quite a complicated website with lots of options, and it can be really helpful for us to show you a little bit about the kinds of things we can explore. So I'd really encourage you to kind of work alongside with this if you can, if you have the screen space and the kind of processing power. Sometimes I know lots of videos and things can make it hard to load things, but you can go to nextstrain.org/ncov/global in your own browser, and this will load up this page, which is the current live build of SARS-CoV-2 that we're hosting at NextStrain. And as the URL hints, this is our global build. We actually run 7 builds every day. We run 1 global build, and then we have builds that focus on specific regions of the world that we can talk a little bit more about if people are interested in this. Now, you might be seeing a slightly different view. If your screen is configured so that you're just seeing the tree right now, then you can use this menu on the left, scroll down to the bottom, and click this box here that says grid. And that means that it will show the tree and the map side by side. And that just makes it a little easier to see how things are changing in both of those as we explore some different options. So again, just at the bottom of the menu, click the grid option here, and you should then see the tree and the map side by side. There's 3 panels on this screen. So we have the tree, the phylogeny here on the left. On the right, we have a map. And what the map is it's plotting kind of the relative sizes of the circles are the number of samples that we have from each location. And at the moment, the circles are plotted over countries. So we can see about how many sequences we have relatively from each country. And if you mouse over a circle, you can see that the corresponding sequences kind of jump up on the tree. Below that, we see the entropy panel, we call it. And I'll go into this in a little more detail in a minute. So first, we'll just focus on the tree here. So the tree right now is colored by region. So you can see this on the map, that every region of the world has 1 color, and this is how the tree is colored at the moment. But we can change this, which I'll go into in just a moment. But the tree is laid out exactly how Richard explained it. So on the y-axis, this is just to keep the branches from being on top of each other, but it doesn't have like a distinct unit or meaning. And on the x-axis, the different samples are plotted through time. So more recent samples are here in September, and more early samples are January, December 2019 here on the left. Now, as Richard showed, we can view this both as the time tree, as we call it, where the samples are plotted on the date they were collected, or on the menu here on the left, we can switch to Divergence View. And this takes us to that mutation view that Richard showed, where now we're showing just the differences that sequences have in the number of mutations. This is a less kind of assumption-prone view because we aren't trying to reconstruct when something might have happened or how sequences are related in time. We're just very simply saying that these sequences have this many mutations the same and this many different. So it's always good to check this view to make sure that we aren't over-interpreting, for example, what we try and infer when we look at the time view, where we have to estimate where sequences are connected back in time. We can do this with different levels of confidence, but it is always an estimate. And when we look at the tree, we can mouse over branches, and we can see that we can get some information about those branches. So we can see how many descendants they have. And often, interestingly, we can see what mutations exist on that branch. So we can see here that there's a nucleotide mutation at position 2881A. And that's led to actually 2 amino acid mutations because it's in an overlapping frame that affects 2 regions, 2 coding regions. So it's changing 2 amino acids. We can also see when we estimate the date for that branch, when that branch diverged. And we can see that we've also tried to estimate what region that this kind of hypothetical ancestral virus was in. So the ancestor here we infer was Europe, but it's always really important to keep in mind that when we make these inferences, we can only use the data that we have. And so we have to be really cautious here. We'll never, for example, infer a location that we don't have a sample from, and we always know that we're missing samples. So we have to be really cautious when we look at these kinds of inferences, what we're associating, how much data we actually have to make that assumption, and how confident we should be outside of the statistical confidence intervals, which is of course mathematical, but also just in common sense. Does this seem reasonable, and do we really think we can make this assumption? We can also zoom in on the tree. So if you click on a branch, my computer is just struggling with all the video, but you can see we zoomed in on a section here, and the map will adjust as well. So we can zoom in a little bit further, and we start to see a little bit more clearly kind of a particular part of the tree. And we can also click on these tips and get more information about a specific sequence. So we can know who submitted it. We can know a little bit about the person that sample was taken up from, for example, their age and sometimes their gender. We can find out some other information as well. If you want to zoom back out again, you can either click on the root branch here, and that will take you back 1 step on the tree. So it will move you back just 1 step. Or if you want to go all the way back out, we can use the reset layout, and that will take you back to the full tree entirely. Just to highlight again, for example, if we zoom in to a kind of interesting looking cluster. So here's a Singapore cluster. It can be really tempting to look at something like this and think, oh man, we know exactly when these transmissions took place. Look, they line up so beautifully on the clock. But this is a good example of why it's always important to check the divergence or the mutation view, because it turns out that we really don't actually have that much signal. We know these sequences are related and we know how many mutations they differ by, but there are probably a lot of different ways this transmission could have passed between different people, and we likely have samples missing. So it's always important to remember not to be too tempted by these time trees and what they reconstruct. It's always a reconstruction, and we need to check the actual data that underlies that inference. So now just to go through a few of the basic features we can use in Auspice, we can adjust the date range here at the top. That's quite simple. We can just, if we want to, for example, focus on sequences from a certain time period, we can drag the sliders in and see that the tree and the map change accordingly to show us what was available at that time. We can exit that using that box at the top. We can also change the coloring. So for example, we can color by country instead of by region. The colors here, there's a lot of colors, so they do tend to still blend together by region, but every country will now have a unique color, though they're not all distinguishable to the human eye probably. And we can color by some other things like age or by sex. We don't have this for all of the data. You can see there's a lot of grays here, but sometimes it can be interesting to look at this. We also have lots of other options, which I won't cover entirely, but certainly some interesting things. Now, one thing that I do want to cover though is that we can color by genotype. So for example, there's a lot that's been said about the D614G mutation. And if we were curious to know, okay, we've heard a lot about this mutation, what does it actually look like in the sequences? We know that this is in the S gene, so we can select the S gene here, and then we can simply type in the position, so 614. And you can see that we can see exactly what's been talked about. The D mutation is the ancestral one that's in these older sequences, and then the G variant has sprung up more recently in these clades here at the top. We can also see, which is quite interesting, how this is distributed around the world. And you can see that, for example, in Asia, especially where a lot of the early outbreak, well, where the pandemic originated, they often have the older kind of mutation. And in Europe, we really mostly saw the G mutation. We can also see that this mutation arose very early on, like in January of this year. So even though the publications relating to it came out over the summer and it got a lot of press attention, this variant has actually been circulating for a really long time. And we are already very familiar with any of its effects, which really counters the claims that a fall wave might be less severe because it has the G mutation. We really wouldn't expect this at all. One more thing, I'll just change back to regional coloring. One more thing I want to show is that we can also change the tree layout. So as Richard said, we can look at different ways of viewing the same tree, like unrooted view. But one that's really important is the clock view, which Richard showed earlier. And this is where we actually plot the date that a sample was taken against the number of mutations that it has. And this is how we can estimate how fast the virus is mutating. And when we do this, you can see we get a very clear line with an estimate of about 22 substitutions per genome per year. So that's about 2 per genome per month. But there is a lot of noise around that line. And that's really important because this is what means that we have limitations in how much we can infer about when things happen. So we can say with a lot of confidence kind of in general terms when things have happened in time, but particularly when we talk about specific events, we need to be really cautious because while there might be an average of 2 mutations per month, there is a lot of variation around this, as you can see. And that means that we don't always know, down to the scale of a week or even a month when something happened. So we need to be, again, very cautious in the interpretations we play here, specifically when this can lead to real life consequences as far as blaming or being convinced that someone or some country is responsible for some kind of spread, when we often just do not have the confidence to say these kinds of things. But it can be really tempting when you look at a colorful tree that has this information in there. So just a couple more things I'll cover quickly. We can change the map view in a couple of ways. So we can change the geographic resolution on the left here. At the moment, things are plotted by country, but for example, we can change to region. And you can see now that there's a big circle for each region. And we can also turn off what we call transmission lines or turn them on. What this is based on that ancestral reconstruction I showed you earlier, where we had kind of inferred that a branch may have existed in the past in Europe. That's what draws these transmission lines. So they have the same caveats that it can be really tempting to look at a sample and say, oh, I can see where this came from, where its ancestors were, how it got transmitted. We have to be really cautious when we make these kinds of interpretations. Finally, I'll just show a little bit of the diversity panel here at the bottom. So this shows just scroll down a little bit more. This shows the entire genome, if I zoom out here, of the SARS-CoV-2 virus. Each of the genes or the coding regions are outlined in different colors. And what's shown here is essentially the number of changes that have happened at specific places in the genome on the tree. And we can show this at the moment, it's in amino acids, but we can switch this to nucleotides if we're interested in changes that also don't affect the coding sequence. And we can do this by what we call entropy, which is kind of a measure of how much that change influences the tree structure, or by just raw events. So just a count of how many times has that mutation taken place on the tree. And if you want to explore these, you can click on any of these. If I can manage to get my mouse. Apparently I can't click because it's too small. Well, sorry, the clicking is not working for me, but hopefully it's working for you. My computer's trying to process too many videos and too many things at the same time, clearly. But hopefully if you're clicking on one of these tips, it's coloring the tree to match and it zooms in a little bit on the view as well. You can do this manually, as I showed you before, where you can select a position. Yeah, my poor computer is just struggling. There we go. So we've zoomed into part of the genome where this is, and we've highlighted the tree by the different nucleotides at this position. And you can put in, I won't try and necessarily do it because the computer's struggling, but you can put in multiple positions if you're interested in looking at different locations at the same time. And then it will show you different combinations between how different different combinations of the nucleotides are spread over the tree. So I think that that covers everything for the kind of basic exploration of auspice. You can find notes covering everything I've talked about here on the website, which I'll post the link again here in a minute. It's not as SARS specific as I've gone into today, but all of the directions, all of the buttons you can press and the different ways you can manipulate are in that map. But I would encourage you to explore for yourself and see what different things that you can see as you play with the tree. And with that, I'll turn over to Anya for the next section.
Just unmute myself now. Okay, I'm going to share my screen and hopefully this will work seamlessly. Share. Okay, hopefully you guys can see that, and I'll just start the presentation. Okay, so I'm going to talk for a few minutes to describe some of these terms: strains, clades, lineages, variants, and explain why they're needed. Why do we need to break down this big phylogenetic tree that we've got into groups? So much time and effort has been invested in genomics for SARS-CoV-2, and it's crucial to get this useful information fed back as soon as possible on a clinical level, and also to governmental bodies, public health bodies, and to these local infection control teams as well. But because we've got 105,000 submissions to GISAID, how do we get useful information out of those sequences, out of over 100,000 sequences? And really, what on the most simple terms you need to do is to define different clusters or groupings, and then you can begin comparing these different groups. And different questions will need different levels of grouping or different resolution. For instance, we see here a schema of some quite large groupings, this overview of the tree, and that could be useful, for instance, to track trends over time. Whereas finer scale groupings, like you see here, might be able to answer slightly different questions. So we have these different terms being batted around that classify viruses at different resolutions. And we hear the term strain quite a lot. And just to clarify, there is only one strain of SARS-CoV-2. Different strains of different biological properties. So to demonstrate what we're talking about in terms of why there's only one strain of the pandemic, I'm going to zoom out a bit. So Richard presented this a little bit earlier, but just looking on this genus level, we see known beta coronaviruses. So we can see, if you can see my pointer, you can see MERS here, and then we can see SARS-CoV-1, and then in red is our SARS-CoV-2, the current pandemic. And if we just zoom into the SARS and SARS-like coronaviruses, again, we can see sort of significant diversity here. Long branches between SARS-CoV-1 and SARS-CoV-2 like viruses, SARS-CoV-2 and coronavirus-like SARS-coronavirus-like viruses. But we can see that there is still a significant level of diversity. Again, if we zoom just into our current pandemic and its closest known sequence, which is a bat virus, we can still see this striking long branch representing about 1,200 mutations. So it's only by zooming into just the SARS-CoV-2 pandemic sequences, which is how we finally begin to resolve the diversity within the pandemic that we're working on right now. So with SARS-CoV-2, there have been over 100,000 sequences generated, but most sequences are within 10 to 20 mutations of one another. So actually grouping the phylogeny is a challenge, but it's crucial to answer questions about the genomics that has been done. In practice, there are currently two different levels or resolutions of groupings that are in use. So we see we've got some clades and lineages as well. So clade systems are sort of large-scale diversity patterns based on SNPs that are across the phylogeny. Things like NextStrain and GISAID have clade systems in place, And if we just focus on the next strain ones, for instance, they have a year letter system with a minimum size and persistence criteria. And this will be particularly useful for long-term tracking of trends in SARS-CoV-2 diversity. Lineages have a different resolution and they're sort of finer scale, and the idea is for them to try and capture the front edge of epidemics. So they might be more useful for outbreak investigations, but it also does mean that they're less stable and that lineages may come and go as additional sequencing is done. Another point to note about the difference between clades and lineages, other than their size, is that lineages have this hierarchy built into them, and also that they use epidemiological information in addition to genetic data. For instance, we define lineages as a potential new cluster that have begun in a geographically distinct place. So we require a SNP to define them because otherwise you can't distinguish one lineage from another. But we also then look for evidence of onward transmission. An example of the sort of quintessential lineage here is given, and we see in this background of UK sequences, we see a distinct lineage where all of these sequences are actually in India, and there seems to be a single branch, so we've identified a SNP, and then we see evidence of onward spread, which roughly translates to diversity within this tree. One thing to note is that size is irrelevant. So you can have really small lineages as well as really big ones. Some of the lineages we've defined are just a handful of sequences, say like 6 or 7 sequences even. But what if you want to investigate at a finer scale, even to the lineage level? Investigate the relationships between 2 different viruses, for instance. And on this level, we need to look at variants or mutations. And this is how we distinguish one virus from the other. And if there are very many variants that actually differ between 2 samples, this gives us usable information. A question that often gets asked, though, is about when 2 viruses are identical. And that sort of brings me straight into the uncertainty and pitfalls section, where we're asking, what is a transmission? And it can be very tempting when seeing 2 identical viruses to say that this proves transmission. However, the question that we can ask from this data isn't what is a transmission, but rather what isn't. If we look at the phylogeny on the left, we see 3 sequences sitting here on what we call a polytomy. So they all lie on the same branch, and based on this tree, we would say that the sequences are identical. And the graph on the right shows an analysis from a couple of months ago where we looked at how far apart in time SARS-CoV-2 sequences can be and still be identical. So how different the sample date can be while sequences can still sit on this same polytomy. And if you see, we actually have some samples that are 90 days apart that still exist on this identical same polytomy. Similarly, we see regularly samples that are sitting identical in the tree from all parts of the globe, particularly early on in the epidemic when before lockdown took place, there was a huge amount of international travel. Now, Richard presented this figure before, but I think it's quite useful to reiterate about what's going on behind the scenes and why we really can't infer transmission. If we look at these blue dots, these are our sampled cases, and the mutations are diamonds along the branches. And all of the red circles are the transmission events that actually happened, but we don't know that. When we have our sampled cases, we use the mutations to reconstruct a phylogeny, and you can see that actually you lose a lot of the information because we don't know what's gone on in between the sample cases. So unless you do have epidemiological information that would suggest transmission, you cannot use genetic data like this to infer transmission. But we can rule out transmission. So if you see something where you have two samples that are very clearly genetically distinct, that belong to, say, a different clade or a different lineage, you can use that to rule out transmission. And we'll look at more of a practical example of that in the session after this. But I'm going to pass on to Emma now for some more cautionary tales.
Thanks, Anya. That's a great introduction. So I'm just going to presentation and then share my screen. And I'm just going to talk a little bit more about some common pitfalls or things that can be tempting to do when you're looking at reconstructed trees. So in particular with regard to ancestral reconstruction. So just to remind ourselves what that is, that's when we know where samples were taken in the world, and then we can try and infer back in time on the tree where their ancestors came from. So here, because we have two Europe sequences near each other, we can infer that their ancestor was probably in Europe. But because we have two North American sequences that are a little more distantly related, we might then infer that the overall ancestor of this totally imaginary virus was in North America. Now, we have to be really cautious though, because of course this is heavily influenced by what data we have and where that's from. So this originally this image and idea was thought up by another member of the Bedford lab, Nick Muller. And essentially what we look at here is, let's imagine that we have some samples from two different countries, an orange country and a blue country. And when we reconstruct a tree from those samples and then try and infer where the ancestor of those sequences is, we might look at this tree and say, okay, the virus started in the orange country and then it spread to the blue country, and there have been 3 separate introductions of the virus from orange country to blue country. But let's say that we didn't have all of these sequences, we were missing a few. So in particular, if we were missing this orange sequence, if that one went away, we now might make quite a different inference. We might say, okay, well, it still started in the orange country and spread to the blue country, but now it looks like actually there was just one introduction in time. We can take that then a little bit further by saying, what if we're missing even more samples? So let's say we only have one orange sample and the rest are blue. We might very easily then assume that this is a virus that's essentially started and spread in the blue country and had one transmission from the blue country to the orange country. Now, of course, the reason that we really need to be cautious is that I've shown this in this direction because it's a little easier to follow. But of course, in real life, we're usually moving in the other direction. We start out with much less information, and as time goes by, we get more samples and we can become perhaps more confident in our reconstructions. But particularly when we start getting sequences or start getting sequences from a particular time point or particular country, we're often in this scenario where we might be missing quite a lot. And so we need to be really careful about how confident we are with what we say and what inferences we're willing to make. And it may be that we never end up getting other sequences and we would never know that we were wrong or not. So we always need to be really cautious, but also not depend on the fact that, okay, well, now it's been a few months and we have more samples. I'm sure we have a complete picture and we can say exactly what happened. We could still always be missing critical samples that might be changing our view on this. So that's just another warning to always be careful, even though we make these beautiful colored trees, to be cautious of what you're willing to say just based on essentially a kind of a branch color that has been inferred just from the data available. One more thing I just want to touch on quickly is often when we make a tree and then, for example, we get some new sequences and we make that tree again, we might have a slightly different tree. And some common questions that we might get are things like, well, where's my sequence? It was in the tree yesterday, now it's gone, particularly on the next strain trees. You might also say, well, my lineage has changed. Yesterday I was getting this lineage assigned, today I'm getting another lineage assigned. Or why is the inference changed? Yesterday it was saying that my virus has an ancestor in Europe, and today it's saying the ancestor is in Africa. What's going on? How can I know what the truth is? But of course, in reality, there's a lot of stochasticity in these processes, which is also really important to keep in mind. So to answer the first question for where's my sequence, certainly when it comes to next strain, we do a lot of subsampling. So as I said earlier, we run 7 builds. I'm actually behind in my sequence counts. We have over 100,000 sequences available now, and we don't use them all in our builds. As you can see, we have just over 4,000 sequences in each of our different builds. So we're subsampling quite heavily from that global available number of sequences. We do this in quite an intelligent way. We don't just randomly sample from that 90,000 to get a distribution over time and through countries, and to sample related sequences when we focus on a particular region, for example. But it does mean that from day to day, the sequences that we pick will change. So we might, pick one sequence from May 2020 from Denmark one day and another sequence from May 2020 from Denmark the next day. So this isn't always consistent across time. That doesn't, these changes aren't meaningful for the tree, but they can be confusing for people. The other thing to remember is, as I said, the trees are really stochastic. So this is just showing on the left, we have a tree from yesterday, or the day before. And then on the right, I have a tree, the same global build, but from August, from about a month previously. And I've used a feature where we can draw lines between sequences that match each other. So the sequences that are the same between the 2 builds. So there'll be a lot of lines that are missing here just because of subsampling, as I already covered. But the other thing you'll notice is that there's been a bit of change in the ordering. These aren't changes that are significant to our interpretation of the tree. They're small changes in how the program has reconstructed little tiny differences. And within the grander pattern, and also if you zoom in, the patterns will have remained the same. But the ordering, kind of whether it appeared, this one on top of this one or this one on top of this one, that will change sometimes from run to run. It almost never, certainly at this point with so many sequences, it never affects our overall interpretation of what's happened. But similarly, this can mean that for the same reasons, your inference as far as where a particular sample, like a very detailed sample, came from might change depending on the sequences that it has in its background in any particular run. Overall, we know the global picture of how SARS-CoV spread quite reliably, but when looking at one particular sequence, this is a lot harder to tangle out. And similarly, the lineage that might be inferred, for example, if you're running pangolin, which we'll cover shortly, it might change depending on the background that your sequence is with over time, but its position at large in the tree is unlikely to change significantly. So this is always important when you're moving from one tree to another or rerunning a tree, to keep in mind that it won't come out the same every time. That amount of stochasticity is expected, and it rarely changes big inferences that we should make. If you are trying to make an inference and finding that it's changing every time you run the tree, it's probably a sign that you need to be a bit more cautious, because if you can't get something to be consistent with different runs, it's probably a sign that the data isn't there for you to be confident in what you're saying. So with that, I'll hand back to Richard. Oh, except for you need to, yeah.
Yes. I hope that gave you sort of a good little overview of what phylogenetics can do, and also what it can do. And despite the fact that we've now spent quite a bit of time warning you of all the things that you're not supposed to do and not supposed to take home, I do hope that you got the idea that yes, there is information in sequences, and that it allows you to sort of group viruses, break an outbreak into different clusters and so forth. I've just pasted a link in the Slack where we've provided, Slack's the chat, where we've provided analysis for pretty much every country in Europe that sort of has a focus on that specific country while sort of providing international context. And you're welcome to explore this for a few minutes and sort of click around NextStrain. I would suggest that we otherwise sort of take a 5-minute bio break and restart at 2 p.m. sort of 14 Central European summertime sharp, with sort of the more hands-on practical bit, where we would like to show you how to analyze your own sequence data with sort of two web tools that our respective groups have developed. Yeah, so that at least was our plan. We'll keep an eye on the chat and answer questions, but I think it'd be useful for you, just take a few minutes to explore NextStrain and all the different features that it offers, either in sort of the global or region-specific builds or these country-specific analysis that we've put up. So see you again in 5 minutes then.
But yes, if you have any question about the first part, feel free to put it in the chat or the Q&A, and we can also answer any burning questions from the first part in this 5-minute break.
Yeah, welcome back, everybody. I hope you had a good sort of click around NextTrain for a little while. So what we want to do in the second hour is give you a bit of a more hands-on experience and touch and feel with the kind of tools that are out there with sequence data, what kind of inferences you can draw, what is available sort of on an ad hoc basis for you to play with. So of course, there's a big ecosystem of phylogenetic tools, bioinformatics, bioinformatic programs and so forth that one can use when one is somebody who does these things every day. But there's also a lot of things that you can do, assuming you know how to interpret the output, with sort of fairly little prior experience. And this is what we are sort of trying to show you a little bit in this second session here. And what we'll start with an introduction to a tool that that's called NextClade, and it's essentially a browser-based tool that allows you to do some rudimentary phylogenetic analysis, plus sort of showing you various features about your sequence data. And if we click on this link here, this is part of our overview page. We should wait. No, that is the right page. You should get a page like this. It lives at sort of at clades.nexttrain.org, and it has sort of a big field here where you can drag and drop a FASTA file, or you can click there to select a file, and I'll do that in a second. Just before we go into this, as it is in the name, NextClade, its primary purpose is to assign clades to sequences that you might have. So you've sequenced a bunch of samples, out comes a FASTA file comes back from your bioinformatician, and the first thing that you might want to ask, sort of, what clade is this? And just to remind you, as Anya discussed earlier, clades are these sort of very large scale classifications of the diversity that we currently see in SARS-CoV-2. And there was a question in the chat again asking about the differences between clades, strains, and lineages. And just to reiterate this, so the terminology of these things are kind of fuzzy, but what we'd like to say is that there's one strain of SARS-CoV-2. And within that strain, we are sort of defining multiple clades, which are sort of a handful of different parts of the tree which share certain mutations, typically without any functional significance. And we define these clades to have a bit of a landmark in this diversity and get some sense as to, is clade 20A or clade 19B taking over, things like that. Is this particular country dominated by clade X or clade Y? And there's currently these two clade systems around. One is by us, by the NextTrain team. And there's a fairly similar one by GISAID, which has a lot of overlap, as Anya showed in her graph. And then there's lineages, and we'll talk more about that later in this session, that are actually meant to zero in on sort of very small groups of the tree and sort of track exactly sort of how one sequence descends from the next and things like that. So what we'll talk about here is clades, which are these more broad brush classifications. One important feature about NextClade here, it's meant to be very simple in that you can just drop your FASTA file on it and analysis will happen. It's also something that entirely happens on your computer. And I'd encourage you to look at, open up this page now in your computer. So it'll load, and all the calculations that we'll do in a second, they will happen on your computer in your browser. Sort of the data that you upload will never actually leave your computer. So you could, of course, you want to continue listen to this webinar, but after loading it, you could sort of unplug your internet and it would still work. You could analyze your data even after you've shut down your internet, only that you won't be able to hear us anymore. But just to reassure you that NextClade is not actually going to send your data anywhere. So it completely happens on your computer. So let me just show you what happens if we analyze some data. So you can either sort of get some example sequences down here, but I'll sort of try to load something that's slightly more interesting. And the link to these sequences is actually available on that page. And I'll take the smaller one of these two data sets. It's a set of sequences that I downloaded just this week from Viper, sort of another sequence database. I'll upload this here, and then a few things happen. A new page opens up, and we'll see sort of an analysis unfolding. And I'll go through the different things that you see now in a little bit more detail. So, of course, here on the very left, you have the names of these sequences. They have some not very interesting names. This is the accession numbers, and then there's the strain name. These sequences are all from the US. And on the very right, you see an overview of the mutations that these sequences have relative to the original, but sort of the reference sequence that was sampled in Wuhan. And at a few positions, for example, this one here, every single sequence has this mutation. It's another one over here, and another two over here. But then there's a whole bunch of mutations that are shared by like either singletons, or maybe they're shared by two sequences. Then there's some that have sort of a pattern of mutation which shows up repeatedly and things like that. Here, sort of, green is a mutation to T, yellow is a mutation to G, blue is a mutation to C, and so forth. Red should be a mutation to A, yes. So you have a bit of a, this should be reminiscent of the alignment view that I showed you in the beginning, where you have some coloring of these of genetic diversity along the genome, right? And this is the very 5' end of the genome. This is the 3' end, and the spike protein sits roughly here. Now you also have these sort of black bits, which in this case are just missing data. This is actually something that is fairly common in these next generation sequencing approaches, that as you try to amplify your sample, some parts of the biochemical procedures don't really work, and in the end you end up with gaps. And this is one of the first thing that next clade will show you here. Yes, you have mutations, but you also have chunks of your sequence that are actually missing, right? And this is one of these sort of sequence quality metrics that NextClade is providing you. So NextClade is, for one, sort of annotating your sequences with a clade. So these all end up being sort of 20 A, C, or B clades. That's why they all share these mutations. But it also provides you with QC metrics, so quality control metrics, that give you some sense of how good or how reliable your sequence looks. And one of these metrics, for example, is a missing data score. In this case, out of a total of 30,000 bases, we are missing about 1,200. And we consider that that's not terribly good, but it's not terrible. That's why it's getting a sort of a yellow for N, the missing data score. It is doing well on the mixed sites. So sometimes in these sequences, you get sites where the sequencer isn't quite sure what it is. So it will report, well, it could be an A or it could be a G. And these have specific codes that you often find in these sequences. And in this site, there is no such sites that we found. But in other sites, in other sequences, you might find sort of 10, 20 or so sites which have missing symbols. One thing where this sequence actually came out fairly not necessarily bad, but at least something to take a close look at. It has a lot of mutations that we don't see in the reference tree. That is sort of a lot of mutations that seem kind of private to this particular sequence, which is often a sign that you want to have a closer look, whether these sequences are real, or whether these mutations are real, or whether there's some sort of sequence assembly, sequence chemistry, sequencing artifact that might generate spurious signal in this sequence. You don't find any clusters of mutations here, which is also a good sign. This is a problem that we mainly had at the beginning of the outbreak, when sequencing technologies weren't, or sequencing protocols weren't quite as advanced. We often saw sort of clusters of mutations, which are often a sign of bad quality. Yeah, then you have a few. The quality score is actually sort of color coded here a little bit. So these are these have some issues, some mutations, which we deem sort of a little bit too many, but most of these mutations, these sequences are actually doing well. There's four which have kind of a lot of private mutations. So that is sort of the big picture overview of what NextClade shows you. But it has one additional feature, and it can show you where these mutations are in the next strain tree. or at least in a slimmed down version of an X-train tree. If you click on this show in tree button, you'll find you'll get a tree like this, where you have a whole bunch of reference sequence. These are now colored in gray, and the new sequences that you just added, which are sort of colored in red. And these, for example, you can then sort of look what is get the details of the sequence. And here you see, this has 14 private mutations that we have not seen otherwise, and that gives us sort of a bad QC score. So these private mutations are actually the mutations that all fall on this terminal branch, which makes this sort of a somewhat questionable sequence. Let's zoom in a little bit more. Yeah, you see that these sequences, they attach to this clade here, but they have a large number of mutations that we've just not seen anywhere else. So this might be real, it might not, but we are flagging this so that the user can take a closer look and investigate the veracity of these mutations in more detail. So as with the usual X-train display, you can then sort of reset the layout. You can also sort of color this tree by various other features. For example, region. The region obviously is not available for your for the sequences you uploaded, because there wasn't any information attached to them, but you still see in what kind of geographic context your sequences fall. So we could sort of go back here. So we see that these sequences that we added, they link up to sequences from the US, but also there's a cluster of South Korean sequences in the same group, some Israeli sequences, Argentina. So as we discussed earlier, the geographic origin of these sequences is often a little bit hard to pin down. Good. So going back to the overall to the output table, you can export this table and additional information to like a tabular file, either in a CSV or in a TSV format. So this is comma separated value or tab separated values, or you could also export it as a JSON file for further analysis with computer programs. You can also filter down these sequences. For example, only look at the bad guys, the mediocre guys, or the bad guys. So there is a filter interface here. You can even filter by a particular mutation. You just have to come up with another amino acid. So you can filter by strains that have a mutation at a particular position. Not sure what's happening. So A6455C. Yeah, so now it works. I just had the numbers wrong. So you can filter by a particular mutation, for example, in this set of sequences, or by name, or by amino acid changes, or by clade, and so forth. Good. Yeah, I was just going to analyze the bigger data set too, which is about 10 times bigger than the one we just did. And I'll just get this started right now. So it's again, you click on this field, you'll get you'll either drop a file there, you click, and you select this file, and it sort of starts doing its thing right away. In this case, it's 161 sequences. And yes, I'd encourage you to do this yourself, either with your own data, which would be ideal, or with the data that we provide. Just have a run, click around a little bit and see what it does. And we'll sort of try to answer your questions in the chat as you go along. And then at some point, we'll hand over to Anya, who will discuss the tools that she and her group have developed. But maybe, yeah, so it will take a few minutes to allow you to actually do this yourself on your computer. And we'll be watching the chat for problems, questions, and things. Okay, so I hope this worked for at least the majority of you, and you got to at least run it, and the details you can sort of continue to explore later offline. We also just stick around a little bit after the session to answer more questions. But yeah, before we do that, I think it'd be good to just cover the lineages and the pangolin tools to give you sort of a full spectrum of classification systems and tools that are around. Anya, do you want to take over?
Yeah, of course. Thank you. I'm going to share my screen again. Okay. And okay, hopefully you can see that. I think now I'm going to just take you through assigning lineages with pangolin, and with an example set that is really quite simple, but I think shows a powerful example of what you can do with lineage information. So all of the material, they're linked from the CoVLineages.org website. I'll just describe a little bit first, but you can navigate over there for the moment if you like, but we'll go there together in a moment. Pangolin is a tool that assigns lineages to a set of query sequences using the lineage nomenclature. So we have Pangolin available as a command line tool, and if you're familiar with the command line, you can do things like configure different settings and quality control levels, and it's also available as a web application. Briefly, Pangolin originally Pangolin 1.0 assigns the most likely placement of your virus sequence into a representative guide tree, and based on that, it determines what the most likely lineage is. So it's very simple. We recently got an upgrade to Pangolin 2.0, which is aka Pangolearn now, and it uses machine learning decision trees to estimate the most likely lineage for your sequence. Again, we've got a sample data set. For the actual practical part of it, you only need the FASTA file, but I'm going to talk you through the CSV file, which has some sort of sample metadata, as if what you'd have in a spreadsheet or something like that based on your samples. So for instance, if you have a set of 10 samples that you've sequenced, and there's an outbreak in a couple of wards in a hospital, you have limited information about where the individuals have been. You might know information that you couldn't share about whether they're a patient or healthcare worker, and you know where the patients were, whether they were in different wards. So based on this metadata, you'd be tempted to assume a number of things like linkage or things like that. So with Pangolin, I've provided a FASTA file. We can download it now, and we can see if genetic information can actually provide another level of information. So I'm going to stop here for a second, and together I'll go to where so on CoVLineages.org, if you haven't reached it already, we can find the Pangolin tutorial here. And hopefully it will load. Cool. So it gives you a little bit of background, and again, you can download the sample data just by clicking on the FASTA file. So I've prepared one earlier. Again, the CSV file is just to give you this sort of scenario information. Now, if we navigate to the Pangolin web application, which is here, you can see we are able to select a FASTA file or just literally drag and drop our FASTA file into that. Now, they've been loaded up. You can see our 10 samples along here, and simple as just clicking Start Analysis button. So each of the sequences gets loaded into a queue, and you'll see the results show up in a moment. Now, one thing to bear in mind is if you're uploading your own data, we don't keep any identifiable information, but when you upload your sequences, they are leaving your local computer and being analysed on a remote server. So if that's something that you can't do, bear that in mind before you start uploading sequence data. But of course, we don't store any information about your sequences. So, okay, so here we can see we've actually got a couple of failed sequences, and we've got 8 sequences that have actually been assigned a lineage. To get a little bit of information about why the sequences may have failed, you can click on the message tab, sequences unable to be processed with Pangolin because this sequence was too short. And likewise, this sequence had an end content of 0.98. So there are certain default levels that Pangolin will just rule out an assignment possibility. So it doesn't want to make inferences about what lineage it is if the data isn't quite up to scratch. But for the sequences that did pass QC, we can see actually we've got 2 different kinds of lineages. So one is just B, and another is B.1.1.65. So they do end up slightly a bit of a mouthful, but are useful to be able to tell apart distinct cases. So we can click on the globe to go into global microreact. So the picture of the UK will just bring you to the UK diversity, because this microreact project has been part of COG UK. But if you click on the globe, it should bring you out. I've loaded a couple from before in case my internet, which is sometimes not very reliable, doesn't work. But if you guys can click on it, you can play around with microreact as well. Yeah, it's going to take a while. So this, while that's loading, here's one I prepared earlier. And we can see the sort of microreact screen. We've got the map showing where the sequence lineage falls. So you can see this. So this is actually the B.1.1.65 lineage. And you can see it's quite a small lineage. There's only a handful of sequences, and they all fall here in the tree. Now, your sequence hasn't been placed in the tree. This is a global tree that's been run daily as part of COG UK, but it does estimate where your sequence would be, because you can imagine your sequence would fall with that lineage. So you can see this is geographically just in the US, and you can get some information, sort of minimal levels of metadata, that's all available publicly, about that particular lineage. And if we open up this one, it's going to have loaded now. So this is the other lineage that are in our samples. And we can see this is much more geographically widespread, sort of many different samples that have been sequenced that are lineage B, because it is one of the basal lineages. And you can see this here where it lies in the global tree. Now, similarly, you can configure all of the different views. If you want to look at the tree in a radial pattern or unrooted, you can do that as well. And you can reposition, so you can see counts of sequences that have been taken from December all the way to now. You can also unfilter. So when you see up here in the top column, this is giving you a lineage view. But actually, if you click this upside down triangle, if you reset all filters, we see this is B, so we're only seeing 1300 of all of the sequences that are currently in microreact. If we reset all filters, we actually get a view of all of the lineages that you see globally, and some pie charts representing the spread around the world. So, and again, dragging these views, you can see the huge number of sequences that have been taken. Yeah, so it's worth playing around and having a click around. I'm not responsible for MicroReact in any way. That is the Center for Genome Pathogen Surveillance. But they and I work together on the Pangolin web app, so this interface that you see here. Wanted to mention as well, you can click this tick box to get a sort of short summary of information about each lineage, and then all of this information can also be downloaded by clicking the download button. You can keep uploading as many files as you like, and with the switch to the machine learning algorithm, actually there's a big speed up now with Pangolin. Before, with the phylogenetic placement, it would have taken quite a while to assign lineages. So I'm going to go back and we can talk about that data then. If I go back to my presentation, hopefully you can still see that. And we can talk about our results and what we got out of that and how that relates back to this scenario of information. So it's a simple example of what these fine scale lineages can do. For instance, so we see two separate lineages, B and B.1.1.65, and you can see that they're spread actually across a couple of different wards, and these two sequences are the ones that had that were too low quality or too short. And with those particular samples, you may need to go back and resequence if you have access to more samples, but it's better to not give the information than to give unreliable information. So simple, but we can see that sample E5 here, which actually is in ward 6 with the rest of these samples. You may be tempted by the metadata to imagine that this is all one linked outbreak, but very straightforward. We can see that sample 5 is actually a different lineage, so it would have a different source of infection. Sample 5 and 10 are the same lineage, but beyond that we can't make assumptions without additional information. Sample 1 is on the same ward as EDB3, but obviously we can't see that now. So sample 1, which is in ward 3, is actually the same lineage as what is in ward 6. And there's two samples that have USA travel history, but beyond that we can't actually link cases. It might be worth investigating further based on what other information is available, but we can only just rule out this linkage. Another tool that I'm only going to talk through because currently it's only available in command line form is LLAMA, which is stands for Local Lineage and Monophyly Assessment. So it's, again, if you have an outbreak example or something like that, and you're looking for finer scale level information, and you want to show relationships between your sequences, you can pull out local trees from the large 100,000 large phylogenetic tree from a set of sequences of interest. And we've been finding this really useful in the UK, investigating outbreaks. So just as an example of how it works, we have the large tree that we identify where the sequence of interest exists. We pull out this local tree. So even though this tree on the right now looks quite big still, it's a lot smaller than 100,000 tips. And then based on the sequences of interest, we summarize the rest of the diversity in the tree. So it really condenses a lot of information into relationships. It can also add in new sequences. If you have just completed a sequencing run and you want to hot off the press see where your sequence exists in that tree, it can add it in. And that means you don't need to rebuild the whole tree each time. And it generates a report, so you get information like this. These are the two sequences that are the two lineages and your sequences of interest that would have been run through Pangolin a few minutes ago. And you see we've got B.1.1.65, and this is actually their relationship to one another, and the two B sequences as well. It also produces a variant graph if you really want to sort of disentangle the relationships between each of the sequences, and summarizes that information in a figure. So from there, I'll sort of give you a few moments if you do want to play around with Pangolin, and I'll stop sharing there. So. And if there's any questions at all, do just add them to the chat as well. I'll see if there's any now.
Yeah, so we've pretty much ended sort of the the formal part of this practical session. Now we are around to answer more questions. We'd also possibly just answer more questions now live as they come in, and maybe sort of just hop in to screen share to address some particular points. One thing, for example, that I forgot to mention is that next Clade also flags mutations that fall into particular sets of diagnostic primers. So if you mouse over a particular sequence there, you should see that some diagnostic primers might be affected by diversity in these sequences. But yeah, please sort of come, play around and sort of put your questions into the chat or the Q&A box, so that we can interact with you guys for a little longer. Also, sort of send us your feedback if you have any suggestions for the tools we've presented. These are all under active development, and the more sort of concrete and specific feedback we get from our users, the better these tools are going to be in the end.
So there's a question in the chat asking to explain the pangolin lineage nomenclature. So I might just share my screen for a second and bring up a figure for that, just to give a little bit of information. Okay. So this figure here, again, I showed it earlier, but I know the lineage system is probably a little bit more complicated than the straightforward clade nomenclature. So this is a phylogenetic tree that you can see here with a set of representative sequences. So obviously not all of the sequences from this current global pandemic, but for the lineage nomenclature, it's hierarchical. So it starts out with a split. A is the base of the current pandemic, and it begins in China, and then from there we see a split into B. So you can see B overlaying here. And this is the first major split, and actually the base of B lies in China as well, with exports around the world. B.1, which is actually, so not quite this long branch, but you can see it's actually the branch just slightly in off this long branch, is roughly equivalent, the base of it is roughly equivalent to the large outbreaks that were beginning in northern Italy and around Europe at the beginning of this year. You can see it's a sort of numbering system, and A, B, and then from the diversity that exists within B, we get these sublineages. So B.1, B.2 is quite a large lineage as well, and so on. From within B.1, the next big jump is actually B.1.1, and we can see quite a lot of sublineages here. So that again continues onwards in the hierarchy. B.1.1 is equivalent to 20B clade. Obviously, these strings of hierarchical numbers can get very complicated. So at a certain level, they do actually create aliases then as well. So a next level from B.1.1.1, we would actually condense that into C. So that's where we get the next step in the alphabet system. And we do have C lineages and we've got D lineages now. So the diversity really has come on, and all of this information is tried to be condensed in this hierarchical system. So hopefully that helps clarify.
There's been some really great questions and answers that have happened during the chat, and we're going to try and export these and get them up on the link that we posted earlier so that you can read through them and learn from the great questions others have answered. And hopefully the links will be a little more clickable as well. I know that's not so easy to get out of the Q&A.
Yeah, so there just was a question regarding the clade schemes, and maybe that is sort of worth pointing out a little bit. Sort of let me sort of pull up this figure. And explain a little, using Anja's slides, explain a little bit sort of how we define these clades and how we are planning on updating them. I just need to sort myself out 1 second. So you should now be seeing the the figure comparing the different clade and lineage systems. And so these here, the center column, these are the next strain clades. On the rightmost column, these are the gizzate clades, which have similar purpose and similar definitions, and sort of have just a slight difference in how things are approached. Sort of our approach was here that we sort of tried to define a new clade whenever sort of a group in the tree, so a defined group in the tree with particular sets of mutation reaches a certain frequency threshold of about 20% and has some global representation. And that we sort of keep adding, capital letters, A, B, C, and so forth, with a prefix which signifies the year in which these clades emerged. This sort of hopefully avoids the problem that other clade systems have, that in the, after some time you end up with sort of A.2.C.X.Y that nobody can remember. So we were sort of trying to encode some context in the sense of when they emerged in time into the name, and hope that we can carry this forward over the years as, if SARS-CoV-2 becomes an endemic coronavirus. Now, how do we define these clades? So they're defined by sort of a set of signature mutations, which are saved in a list that somebody asked about in the chat. And sort of what our program does, it sort of essentially walks up the tree from the root and sort of finds the first place in the tree that matches this clade definition. And then everything downstream is going to be, that clade, unless there's another clade definition nested within this. And this makes this, the clade assignment on the tree robust to repeated mutations at the same position. Because, that internal node that defines the root of this clade will still have these mutations, even if some of its children have mutated at this position again. So we'd expect this to be reasonably robust to recurrent mutations, and this is something that we've used for influenza clades over the years, and it has worked quite reliable for that purpose.
And I've just posted a couple of links in the chat. So there's one to our NextStrain discussion board. So if you are exploring NextStrain and end up having more questions about how things work, this is a good place to post, or to probably look first and see if someone else has posted the question. If not, to ask your own question, and it can be answered by other people who use NextStrain or one of us on the NextStrain team. If you're interested in getting more hands-on, I've also linked to our GitHub repo that actually runs all of these builds. It's all open source code, and you can see how it's done. There's also a link in the README on that repo where you can follow instructions to set up your own build. There's a link to a tutorial that will help you do that. This is a little bit more involved. It's good if you have some programming and command line experience before you try this, or to maybe get some help from someone who does. But if you are someone who is a bit more comfortable with that, then you might be interested in giving that a try if you have your own data or want to set up a custom build for a country or a region or a district that we don't already have.
Okay, we still have 45 participants in the webinar, so yeah, keep your questions coming.
So Rabindra asked a great question of whether we can just use the software for SARS-CoV-2 or not. The repo that I linked, the one that the /ncov novel coronavirus before it had an official name, that one is very SARS specific and assumes you're running SARS-CoV-2. But the NextStrain website and software in general is very flexible. And it was actually developed a long time before we knew SARS-CoV-2 was going to come to our humble planet. And we've used it on many, many pathogens. So it was originally designed to work with flu, and you'll find lots of resources for flu builds. If you just go to nextstrain.org, you'll see many of the different organisms that we've run and that we maintain. So we've done flu, we've also worked with Ebola, with Zika, with different enteroviruses, tuberculosis, and some collaborators and other people have used NextStrain for even more diverse organisms and pathogens than that. Some of those are also linked on the nextstrain.org website. And now we don't often have as in-depth tutorials for these, unfortunately, because there's been much more interest in SARS-CoV-2 than anything else this year. But all of that is open source, and you can usually follow links to those repositories and see how that was done if you're interested in replicating that on a different pathogen or a different virus that we haven't covered. And we're always interested to know how people are using NextStrain, both on SARS-CoV-2 and on other organisms, what you find it useful for and how it's working for you, because we're always very interested to see how other people use the software.
And just to follow up with that,
yeah.
Pangolin is, again, SARS-CoV-2 specific. It assumes that you're assigning a SARS-CoV-2 genome. It actually won't assign anything if you try and give it a sequence that's too divergent. So it will just come up again with one of those little errors. But Llama isn't virus specific or organism specific. So if you have a background of diversity, of phylogenetic tree that you can supply, Llama can pull out your sequences of interest and summarize that information for you. So that's not specifically just for SARS-CoV-2.
Yeah, Wolfgang asked about Pangolin 1. That's probably also for you, Anya.
I maybe can clarify about Pangolin 1. I'm not sure what the question means, but Pangolin 1 is also SARS-CoV-2 specific. You could, in theory, give it a set of a tree sequence of representative trees with a hierarchical nomenclature structure, and it would assign the most likely nomenclature system. I've only ever tested it in SARS-CoV-2, but actually all of the code is up on GitHub, and it links from the website covlineages.org, and you can look at the code if you're interested in trying it out with different data. That it definitely could be possible to be used. I just haven't tried it. Who knows? You might discover something. So yeah, but it specifically would need the system to be hierarchical in order to infer the most likely lineage. And also Pangolin 2 is infer based on training, a training set, and uses a specific model that gets loaded into the environment when you're running Pangolin. So in theory, you could train Pangolin with this sequence on different data, and maybe in the future that's something that we could look into adapting it for.
Yeah, there was one question regarding viral quasi-species. So we at NextStrain, we haven't sort of ventured too much into intra-host diversity because, well, for acute infections like influenza or SARS-CoV-2, there typically isn't a hell of a lot of diversity shake a stick at. For other pathogens like HIV, you typically get a lot of intra-host recombination, and then sort of this whole idea of building a tree is no longer that good of a guiding principle. So there have been attempts to sort of try to incorporate information from superinfection maybe, and sort of intra-host SNPs that go across transmission bottlenecks, but NextStrain currently doesn't offer this. Okay, sort of more questions for us?
And the recording of this should be available shortly for people who registered for the conference. So if you have colleagues or friends who you think might also be interested but couldn't make it today, or now that you've seen it, you think it might be relevant for them, feel free to pass it on and they can watch this all at their own convenience, the video. Yeah, there's one question regarding recombinant samples. Yeah, you typically find that these samples, if you took that sequence and split it in half, you typically find that the first half of the sequence wants to be in one place of the tree, and the second half wants to be in another part of the tree, and that is sort of a telltale sign of recombination. Of course, where in the sequence this breakpoint happens varies. We have not seen sort of very good examples of that for SARS-CoV-2 yet, even though it is conceivable, because coronaviruses do recombine quite a bit. They often tend to recombine in sort of kind of limited sections, not like half genome chunks. But so far, we haven't seen much of this yet. But we have a NextStrain sort of a way that Emma showed earlier to compare two trees side by side, so-called tanglegrams, and that's where you'd often see signatures of recombination. So there is one question about cycle threshold. So CT value thresholds for sequencing. Yeah, that depends a lot on sort of how good your PCR is. And if you have very high CT values, you'd probably get gradual dropout out of amplicons, because some of them work better than others. But yeah, sort of typically, I think sort of in the low mid 30s is a typical cutoff, and it gets really tough in the high 30s. But we are probably not the right people to ask this, because we are not actually doing the sequencing. Right? we are just taking the output and trying to analyze it.
I've just linked in the chat to the Arctic Network, what was called when it was made ENCOV 2019, or SARS-CoV-2 resources page on the website, and that has both wet lab and dry lab recommendations So there's a sequencing protocol there. It links out to protocols.io, and it has various CT recommendations on what is a good and what isn't a good enough sample to do nanopore sequencing on. So this is specifically that platform. I know other people have adapted it to Illumina, but that might be somewhere you could find that information.
So there's one additional question regarding a sequencing read with specific mutations. So, that if it's a single, you should never base anything on a single read. So most of the SARS-CoV-2 analyses are sort of based on consensus sequences. And for a small viral genome like this, you'd hope that you have coverage of hundreds to thousands. And if a single read has multiple differences to the rest of the reads, then that's probably just a problematic read.
And like what Richard mentioned earlier, even when you do see consensus sequences that have a lot of unique independent variants, that is also even flagged as maybe a problematic sequencing run or sample or something like that, if there are too many unique sites, more than you'd expect from a particular sample taken at a particular time.
Yeah, in particular, the bioinformatic cleanup of the reads can, if that's not done properly, that can result in a lot of spurious diversity. For example, sort of primers that are not properly clipped from the amplicons might sort of make you think that your sequence is actually different from what it is. So if wobble sites and primers might sort of give you diversity that isn't actually there. So there's sort of a whole bunch of bioinformatic pitfalls too, which we haven't covered here. But that's partly what sort of the next Clade quality control assessments are meant to help you with. Not all, it doesn't catch all of the problems, but it hopefully catches some. Okay. So I hope this was useful to many of you. As I said before, sort of if you end up using these tools that we presented, do let us know what your experiences was, what you think would be useful additions and things like that, either via sort of issues on GitHub or via email to us directly. I don't really have much more to add. Thank you all for attending and listening, and all the great questions that kept this sort of pretty interactive, which I think we very much enjoyed. Wish you a good rest of the conference, and have a good day. And Daniele, I don't know, Emma, Anya, and Daniele, do you want to say something before we close, or otherwise we'll just ask Daniele to close it?
No, I think it's been a great session. Thanks, everyone. We had some really brilliant questions and answers, and that always makes these things so much more interesting and engaging. So thank you for the great questions, and we hope this is really useful and helps you go forward with confidence when you encounter sequences in the future.
Yeah, just to reiterate that as well, really nice discussions, and thanks very much.